Welcome back to Cool Medicine. Today we're going to talk about glycolysis step by step. Glycolysis is the process of breaking down one molecule of glucose into two molecules of pyruvate. This process takes place in the cytoplasm and is absolutely essential for every cell, especially red blood cells, which have no mitochondria. So we begin with glucose. There is one enzyme called hexokinase, or its version in the liver called glucokinase, that adds a phosphate group to glucose, making it glucose 6 phosphate. The process of this step is to trap glucose inside the cell. This costs us one molecule of ATP. We can consider it as an investment. Next, the enzyme phosphoglucose isomerase rearranges glucose 6 phosphate into fructose 6 phosphate. Nothing's gained or lost. It's simply a structure rearrangement. Now comes the most important control point of glycolysis. The enzyme phosphofructokinase 1 adds another phosphate group from adenosine triphosphate, converting fructose 6-phosphate into fructose 1,6-bisphosphate. This step uses another molecule of ATP. It's known as the commutative step of glycolysis because once fructose 1,6-bisphosphate is formed, the cell is committed to continue through glycolysis. Hence the name kinase, we use 1-ATP. Fructose 1,6-bisphosphate is then cleaved by an enzyme called aldolase into two 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The enzyme triphosphate isomerase converts dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate. From this point forward, every reaction happens twice because we now have two molecules of glyceraldehyde 3-phosphate. Each molecule of glyceraldehyde 3-phosphate is oxidized by the enzyme glyceraldehyde 3-phosphate dehydrogenase into 1,3-bisphosphoglycerate. During this reaction, NAD is reduced to its reduced form, NADH. Since there are two molecules of glyceraldehyde 3-phosphate, we gain two molecules of NADH. And we put one phosphate group. So now we have two phosphate groups in 1,3-bisphosphoglycerate. Next, each molecule of 1,3-bisphosphoglycerate donates a phosphate group to ADP, producing ATP. This reaction is catalyzed by the enzyme phosphoglycerate kinase, and it produces two molecules of ATP in total because obviously we had two glyceraldehyde 3-phosphate. And the result molecule is 3-phosphoglycerate. Now, 3-phosphoglycerate is then converted to 3-phosphoglycerate by an enzyme called phosphoglycerate mutase. It's just rearrangement, not a big deal. Then the enzyme enolase acts on 2-phosphoglycerate, removing water and forming phosphoenolpyruvate. This molecule is very high in energy, ready to give away its phosphate group. Now comes another important energy producing step. The enzyme pyruvate kinase transfers the phosphate group from phosphoenolpyruvate to ADP, producing pyruvate and another molecule of ATP. Again, because we started one glucose molecule, this happens twice. We make two more molecules of ATP. So let's summarize glycolysis. Glycolysis starts with one glucose molecule and ends with two molecules of pyruvate because we had two molecules of glyceraldehyde 3-phosphate. Along the way, we use two molecules of ATP, but we make four molecules of ATP, giving us a net gain of two ATPs. We also produce two molecules of NADH, which carry high energy electrons. Now, what happens to pyruvate depends on the oxygen conditions. Under anaerobic conditions, meaning without oxygen, pyruvate is converted to lactic acid by an enzyme called lactate dehydrogenase. This reaction regenerates NAD in its oxidized form because it's necessary for glycolysis to keep running. Under aerobic conditions, meaning with oxygen, pyruvate is transported into mitochondria where it's converted into acetyl-CoA. This molecule then enters the Krebs cycle to produce much more energy. So, glycolysis gives us a quick energy, but if oxygen is available, the cell can go much further, extracting a huge amount of energy through the Krebs cycle and the electron transport chain. So, that's it for today. If this video intrigued you, don't forget to like and subscribe, and join me next time as we uncover more mysteries of medicine. See you next time.